Sporophytes are the adult plants that you see, and they are diploid. Gametophytes are haploid, and only mosses, really, that I know of are gametophytes as adults or haploid. And we discussed the reproductive structures of flowers. So there are 4. I'll put up the chat. What are the 4 reproductive structures or floral structures of a flower? What is the name of those 4 floral structures? Four floral structures or organs, 4 floral organs. You can start with petals. What else is there besides petals?
The stamen?
Yes, the stamen, good. Yeah. Another one starts with C, the females. What are the females called, the female parts? Starts with a C. Carpels. So stamens, carpels, male, female. Petals. And then there's something else starts with an S underneath. And those are sepals. So remember that a complete flower has all 4. Has all 4. And we looked at the development of the sporangia. So the microsporangia in the male parts of the flowers and the megasporangia in the female parts of the flowers, which produce eggs and sperm. The male also at the end of meiosis and mitosis produces a generative cell and a tube cell, and the female produces, the megaspore produces antipodal cells, 2 polar nuclei, 1 egg, and 2 synergistic cells that will direct the tube to the ovary when the pollen tube starts to grow down the style. So now we are moving on to what happens after fertilization. So the pollen tube is formed, it's moved down the tube, attracted by calcium or another chemical that's exuded by the synergistic cells, and then the sperm travel down that tube. One of the sperm get together with the 2 polar nuclei, and then that eventually becomes endosperm. And the other sperm fuses with the egg, and that becomes the embryo. And then after that, the ovules develop into seeds and the ovaries into fruits. So after landing on the receptive stigma, pollen grain germinates. It's called germinating. It has to fill with water in order to germinate usually. Pollen grains are very tough, so the water helps to soften it, as well as enzymes. Yeah, and then the pollen tube discharges 2 sperm into the embryo sac. And with double fertilization, 1 sperm fertilizes the egg and the other combines with the polar nuclei, giving rise to the food storing endosperm, which may or may not get passed on to the cotyledons, possibly get passed on. So what's the point of double fertilization from an evolutionary perspective? What is the advantage? It appears the advantage would be that The endosperm will develop only in ovules where the egg has been fertilized. So the endosperm of the plant, of the flower, um, it's quite expensive to make for the flower. It contains protein and lipids and carbohydrates. It contains the nutrients that the embryo is going to need to grow. So it's a waste to invest energy in the nutrients if you're not even going to have an embryo. Yeah. So this prevents angiosperms from, I love this word, squandering, recklessly making or recklessly spending nutrients in eggs that lack an embryo. Yeah. So the growth of the pollen tube is quite interesting. Um, it has been shown that in the reproductive structures, once a pollen grain lands and the tube starts to grow, the reason for the growth is probably due to cytoplasmic calcium. So I wanted to say a couple of things here. One of which is, this is the process. I'm not going to go through every step because we've talked about it already. So this is just the diagram showing the steps of fertilization and growth of the pollen tube. But what I wanted to say is very interesting, which is that after fusion, after gamete fusion, there is a block to polyspermy. So in case other pollen, which other pollen will probably have landed on the stigma, and maybe they're growing a pollen tube as well, and maybe they're going to release sperm as well, but those sperm are prevented, just like in animals, from fertilizing or trying to fertilize the same egg. Plants establish a block to polyspermy, or more than one sperm getting to the egg. So from ovule to seed. So after double fertilization, the ovule develops into seed, the ovary into a fruit. I have a feeling we've said that before. The development of the endosperm precedes embryo development. So the egg has been fertilized, but the endosperm starts to develop first. And the endosperm is a stockpile of proteins, oils, and starch. So in most monocots and some dicots, it's often called eudicots now, the endosperm stores nutrients that can be used by the seedling after germination. But in some eudicots, the food of the endosperm is completely exported to the cotyledons. So after double fertilization, The sperm has combined with 2 pollen nuclei, so now it's a triploid nucleus. It's in the center of the ovule. It starts to divide. So it's the nucleus that is dividing, the triploid nucleus that is dividing because it's fused, and it forms this giant multinucleate cell called a supercell. That has a kind of milky consistency. And then through cytokinesis, the cell starts to form membranes between the various nuclei. And cell walls form and the milky consistency becomes solid. Now the embryo will develop as well, of course. The first mitotic development of the zygote is transverse. So here we are, the zygote. It's transverse and it forms a terminal cell and a basal cell. The basal cell becomes, I have to check this to make sure I've got it right. Yeah, the basal cell is the part of the embryo that will implant into the ovule, into the plant itself, or into the seed, I should say. And the suspensor will grow into the embryo. That's a terminal cell. So the terminal cell grows into embryo, and the basal cell implants. Yeah, or anchors. It anchors itself into the parent. And in that way, the nutrients can be transferred. So here's the suspensor that's anchoring the cell. The part that will become the roots, the apex of the root, the shoot, and the cotyledons. This is the seed coat, and this milky area here is the endosperm. So the seed itself, where the embryo is encased, and its food, the embryo and its food supply are enclosed by a very hard protective seed coat. And during the last stages of maturation of the seed, a seed dehydrates. So it will lose most of its water. And then the embryo stops growing at this point until the seed germinates. And then it'll start growing again. And the seed coat is formed from the integuments. I don't know if you remember those, but there are some cells that were part of the ovule, the outer part of the ovule. So here's a common garden bean, a dicot or eudicot. The embryo consists of the hypocotyl, hypo means below, the epicotyl, the radicle, and the cotyledons. So this is the cotyledons here. So that's where this seed, the embryo, where the embryo will get its supply of food. Castor beans are similar, but they have very thin cotyledons. And instead, the embryo absorbs food from the endosperm. A monocot has a single cotyledon, hence the name monocot. It also has a coleoptile and a coleorhiza. So this is the coleoptile, the coleorhiza. And what's very interesting is that the coleoptile is an encasement as it grows in the soil so that it doesn't get damaged by the soil and the friction of the soil. So a fruit develops from the ovary, and the fruit has a couple of functions. It can protect and close seeds. It also aids in the dispersal of seeds by wind or animals. And fruits are classified into lots of different types. It depends how they develop. So here are peas. Peas are a fruit, a simple fruit, which develops from a single carpel or several fused carpels of one flower. So these are from fused carpels. They're simple seeds. An aggregate fruit is something like a raspberry, develops from separate carpels of one flower. So the flowers of the raspberries and the strawberries and the blackberries, they have lots of carpels. And an inflorescence forms a multiple fruit, which develops from many carpels from many flowers. So separate flowers on the same plant. So these segments all came from separate flowers on your pineapple. So how does the seed germinate? At first it's dehydrated and it passes into dormancy. And that's very important because there might not be the right amount of water or the right amount of nutrients, or the seeds might not be in a position to germinate because of temperature. But it's a good thing because it increases the chances that germination will happen when it's good, when times are good, at a time and place that's advantageous. Yeah, in particular, away from the parent because he doesn't want to be competing with the parent for food. So to break that dormancy, there must be environmental cues. So often it's temperature. So they'll germinate in the spring, for example, when it gets to be warmer, or lighting cues. So that's an example of photoperiodism. And then they grow. So if they germinate, which they do with imbibition, that's the uptake of water. So water is required to burst that hard seed coating. And the radicle is the first thing to emerge. So once the seed is germinated, the radicle emerges and that becomes the root. In some dicots, the hypocotyl forms this kind of hook and it pushes pushes the growing embryo and the seed above ground. Monocots have a different method. This is the coleoptile. So the coleoptile itself is surrounding the embryo, and that new leaf, which is very tender, pushes upward through the soil and into the air, and then it can start to photosynthesize. So that was all about sexual reproduction. Of course, there's lots of methods of asexual reproduction. But sexual reproduction does produce genetic variation. So without genetic variation, there isn't anything really for natural selection to select from. Asexual reproduction in plants is also called vegetative reproduction. And there's lots of methods. Fragmentation. So the parent plant might just be fragmented into parts that develop into whole plants. That's pretty common. So you can take cuttings from plants, for example, or bits of plants can break off and they may grow into whole new plants if the
Like this would be
conditions are good.
hedges and stuff we producing, pardon me.
Sorry?
Hedges we put, like hedges would we produce by this manner, right?
Yeah, hedges could reproduce by this manner. Yeah, for sure. Yeah. Sometimes the root system of a single parent gives rise to what are known as adventitious shoots, and they all become separate shoot systems. So this is a grove of, I think they're aspens, and they all have the same root system, the same parent. Indeed, the oldest living organism, as far as I know, is 80,000 years old. And that's a root system that's existed for that long. The, the trees themselves, they have gone through lots of things and died off, but the system maintained intact and about 80,000 years old. And of course, for agriculture, asexual propagation is important because it's very fast. Plant fragments that we can get from plants are called cuttings. I don't know if you've heard of grafting. It's used a lot in the wine industry. You can take a twig or a bud from one plant and graft it kind of with glue, essentially, onto a plant of a closely related species or a different variety of the same species. So with vineyards, for example, the roots and the stems of North American vine plants are really hardy, but the European stems and fruits are really delicious. So they graft them together. They graft the delicious fruit producing parts onto the root parts. It's called grafting. You can clone them in vitro methods. This is an example of just a few parenchyma cells from a carrot gave rise to this callus, which is a mass of undifferentiated cells. And then if you provide the right medium, they will differentiate. The callus will differentiate into an entire plant with all of the different organs. A protoplast fusion, you can fuse a protoplast. Protoplast is the name given to cytoplasm of plant cells. So they remove the cell walls and they create hybrid plants that way. So plant biotechnology transforms agriculture. As you know, we've talked about this before, biotechnology and genetic engineering. It refers to innovations in the use of plants to make products for humans, and it also refers to genetically modified organisms in agriculture and industry. So genetically modified can occur with selective breeding. And we've been doing that for 30, 40,000 years. We've selectively bred plants. So the plants that you see in the grocery store are unlike the originals. They're genetically completely different. And the other type of genetically modified organism is by genetic engineering. And that's where you can place the gene from one species into another species. And that's very useful in a lot of cases. Artificial selection, of course, where humans select the genetic makeup. We've been doing it for thousands of years. Maize is a product of artificial selection, but it's not a great source of protein. Hybridization of plants is pretty common in nature, so it's used by breeders. Are we trying to reduce world hunger and malnutrition? Well, there's different methods of doing that. We talked about this before, I think, a little bit, didn't we? The golden rice method to promote, I think it was vitamin A in countries where vitamin A is deficient. Some biologists seem to be concerned with unknown risks associated with GMOs. Some are concerned that engineering genes may transfer allergens from a gene source to a plant used for food. So the allergens and other risks have not been seen through scientific studies. So far, there haven't been any risks materialized in the studies of GMOs. One thing is that the modified crops may have effects on non-target organisms. Don't really know how that would happen, but perhaps the pollen from one plant could land on a stigma of a different plant. Some scientists think that introduced genes may escape from a transgenic crop into a related weeds through crop to weed hybridization. I haven't seen studies like that. But there are enormous benefits of genetically modified crops. Without them, we literally would not have enough food for the world. So that's where I'd like to end. Thank you for watching.